What I would like to share with you today is some recent and unpublished work where we're trying to understand how cardiac energy, or cardiac insulin resistance, occurs in the failing heart. As you all know, that glucose is a major contributor to cardiac ATP production along with fatty acid, and that insulin stimulates glucose oxidation by enhancing glucose uptake and glycolysis, and thereby enhancing pyruvate supply to the mitochondria. Insulin can also, through an unknown mechanism, directly stimulate glucose oxidation independently of enhancing glucose uptake and glycolysis. And this mechanism seems to be mediated through the activation of pyruvate dehydrogenase, which is the rate-limiting enzyme in mitochondrial glucose oxidation. What happens in the failing heart is that we see a marked cardiac insulin resistance, and this is associated with impaired cardiac insulin signaling pathway. So, for example, if you look here, these are myocardial biopsies taken from patients with heart failure, and you would appreciate that there is an increase in the phosphorylation of the inhibitory sites of insulin receptor substrate, along with decrease in the phosphorylation of AKT and GSK-3 beta. So it's clear that these hearts are insulin resistant. And this cardiac insulin resistance is mainly manifested by a marked reduction in insulin-stimulated glucose oxidation rates in these failing hearts. So for example, if you stimulate a heart with insulin, you see this nice increase in glucose oxidation, which was blunted in the failing heart. And this insulin resistance was associated with a decrease in the insulin signaling pathway in these failing hearts. So as a compensatory mechanism, the failing heart becomes more glycolytic with an increase in glucose uptake to compensate for this reduction in the overall mitochondrial oxidative metabolism. However, these high rates of glycolysis are not accompanied with a comparable increase in mitochondrial glucose oxidation. And we often refer to this as an uncoupling between glucose oxidation and glycolysis. So we ask specific questions. How is insulin signal transduced from the cell membrane to the mitochondria to directly stimulate glucose oxidation? And why this direct stimulation of glucose oxidation impaired in the failing heart? And whether or not it's possible to rewire this mitochondrial insulin signaling in the failing heart. So we know that AKT, GSK-3 beta, and PKC delta are major components of the insulin signaling pathway. And it's been suggested that these kinases can be translocated to the mitochondria following insulin stimulation. So we ask the question whether this translocation of, or the translocation of any of these kinases could possibly mediate, at least in part, the direct insulin stimulation of glucose oxidation or not. So to address this question, we randomized two sets of male and female C57 mice hearts to be perfused for 30 minutes in the presence and absence of insulin. And by the end of the 30 minutes, we took the hearts from both sets, and we isolated the mitochondrial and cytosolic fraction to look at the translocation of these kinases. And what we saw is, in the absence of a significant change in glycolysis, we see this nice stimulation of glucose oxidation when we stimulate this heart with insulin. And this stimulation was associated with the translocation of AKT, PKC delta, and GSK-3 beta to the mitochondria. What we also saw is that this translocation was associated with dephosphorylation and activation of PDH, which is aligned with this nice stimulation of glucose oxidation. So we wanted to know next whether this is just an association or indeed the translocation of any of these kinases is mediating the direct insulin stimulation of glucose oxidation. So to do so, we use the same isolated working mouse heart model, but this time we perfused with specific inhibitors of these three kinases. Namely, we used LY29 and AKT8 to inhibit AKT. We also used bisindol to inhibit PKC delta, and we used 3F8 to inhibit GSK-3 beta. And again, at the end of these perfusions, we took the heart and we isolated the mitochondrial and cytosolic fractions. So what I'm showing you here is the glycolysis rates, and you would appreciate that there was no significant difference between glycolysis rate with any of the pharmacological inhibitors, which suggests that we didn't have a significant effect of any of these pharmacological inhibitors on neither glucose uptake or pyruvate supply to the mitochondria. However, when we look at the glucose oxidation, again, when we stimulate the heart, we see this stimulation of glucose oxidation, which was completely blunted by either of the AKT inhibitors, suggesting that, in fact, the translocation of AKT to the mitochondria plays an important role in mediating the direct stimulation of glucose oxidation by insulin. What was also interesting to see is that when we inhibited PKC delta, we saw that the basal rates of glucose oxidation were increased in the absence of insulin, and when we add insulin, we didn't see further increase in glucose oxidation, which possibly could be due to the maximized effect that we achieved when we inhibited PKC delta. While when we inhibited GSK-3 beta, we still see this nice increase in glucose oxidation, suggesting that maybe GSK-3 beta is not part of this mechanism. Consistent with that, when we looked at the fatty acid oxidation, again, when we stimulate the heart with insulin, we see this nice inhibition of fatty acid oxidation, which was again blunted when we inhibited AKT with either of our pharmacological inhibitors. And when we inhibited PKC delta, we see that the basal rates of fatty acid oxidation were inhibited, and when we hit with insulin, we don't see further inhibition. And again, inhibition of GSK-3 beta, didn't have much of an effect on the direct stimulation of direct action of insulin inside the mitochondria. So we went further and we wanted to characterize the localization and the translocation of these kinases inside the mitochondria. And we were really excited about what we found here. We found that when we inhibited AKT with either of these inhibitors, we saw that the mitochondrial AKT was also inhibited. What we also found is that when we inhibited PKC delta, we saw an increase in the activity of mitochondrial AKT. So taking all of these findings together, this suggests that the mitochondrial AKT is a prerequisite to mediate the direct insulin stimulation of cardiac glucose oxidation. So what we wanted to investigate next is, does selective enhancement of mitochondrial translocation of AKT mitigate cardiac dysfunction and or adverse remodeling in the failing heart? So to address these questions, we randomized two sets of male and female C57 mice to undergo either sham surgery or transverse aortic restrictions to induce heart failure. And 3 weeks after the surgery, we did an echo to assess cardiac function. And at this stage, we randomized animals with the same cardiac function to either receive vehicle or SC79, which is an AKT activator, for 2 weeks. And by the end of the treatment protocol, we did another echo to assess cardiac function and cardiac hypertrophy. And hearts from all of these groups underwent the standard isolating working heart protocol. And then we took these hearts and we isolated the mitochondrial cytosolic fractions to look at the translocation of AKT in these hearts. So what I'm showing you here is some echo analysis by the end of the 3 weeks post-TAC, and you would appreciate that animals in both sham groups and the TAC groups had similar ejection fraction as well as LV mass by the end of the 3 weeks post-TAC. At this stage, we randomized these groups to either receive vehicle or SE79. And what we saw by the end of 2 weeks treatment is that animals that receive vehicle continue to have a reduction in cardiac function, as well as an increase in LV mass, which is an indicative of adverse remodeling. However, when we treat with AKT activator, we see this nice improvement in ejection fraction, as well as limitation in the adverse remodeling. And we were really excited and happy about these data and about this dramatic effect with only specifically targeting the AKT in the failing heart. So we wanted to characterize the metabolic impact of these treatments. And again, when we look ex vivo, we see the same cardioprotection in these hearts that receive AKT activator. Looking at the glucose oxidation, you would appreciate that the failing heart had a reduced insulin-stimulated glucose oxidation. And when we activate AKT, we see that this was significantly improved. So we next ask the question, what happens to the mitochondrial AKT and whether the translocation was affected in the failing heart? So here we saw that there was indeed a significant reduction in the mitochondrial translocation of AKT that was associated with an increase in the phosphorylation and deactivation of PDH, which is consistent with this reduction in insulin-stimulated glucose oxidation. However, when we treat with SE79, we see that an increase in the mitochondrial translocation of AKT that was also associated with dephosphorylation and activation of PDH, which is again consistent with this nice increase and improvement in insulin-stimulated glucose oxidation. So to sum up, I hope that I was able to convince you that insulin directly stimulates mitochondrial glucose oxidation independent of enhancing glucose uptake or glycolysis, and that mitochondrial translocation of AKT is a prerequisite to mediate this direct stimulatory effect of insulin, and that inhibition of mitochondrial translocation of PKC mimics insulin's action on cardiac glucose and fatty acid oxidation, and stimulating mitochondrial translocation of AKT attenuates cardiac dysfunction as well as adverse remodeling in the failing heart. So just to put things in perspective, this discovery, we believe, provides valuable insights concerning the potential of selectively targeting mitochondrial translocation of AKT and or PKC delta to overcome cardiac insulin resistance in patients with heart failure, as well as other conditions that are associated with cardiac insulin resistance, such as diabetes and obesity. And I think I'm going to stop here by thanking everybody in the lab and people who contributed to this work, along with the collaborators and the funding bodies. And thank you very much for listening.
Okay, so I would like to thank all the three speakers to finish our session on time. First, thanks to Qutuba, then Zhao, and then finally Chris. Let's start our question answer session. The first question is from Helen Collins for Qutuba. So she's wondering whether did Qutuba notice any sex differences in terms of insulin treatment or TAC or targeting AKT in Qutuba's experiment, or these are data pooled, whether Qutuba used male and female mice together to generate these data?
Yeah, so thank you, Helen. This is a really important question. I'm sorry I didn't have time to.
So sorry to interrupt you, Qutuba. I think your camera is switched off.
Oh.
So you may want to. There we go.
Yeah, I'm sorry. I was on the phone.
Sorry.
Sorry for that. Yeah, excellent question. So the main reason that we started the first series of experiment to use male and female is to look if there's any significant differences between the male and female in either the cardiac energy metabolism in general, namely glucose oxidation and fatty acid oxidation. And by comparing the data, we didn't see much of a difference between male and female in terms of the translocation of these kinases into the mitochondria. So we can see the same pattern of translocation with male and female. Also, there was no significant difference between cardiac energy metabolism in terms of glucose and fatty acid oxidation and glycolysis as well. So in the next, well, with the next series, we just decided to continue using both as far as they are both physiologically relevant and there's no significant difference. And then we pulled all the data together. So yeah, so the little secret here is started with male and female, thinking that there might be some differences, but no, after the experiments and analyzing data, we pulled them together. Yeah, good question.
Thank you. The next question I'm seeing is for you, Qutuba, as well. It says, were there any changes in GLUT4 due to altered AKT signaling?
Yeah, that's I think Joe Sun is asking about what is the site of action for the phosphorylation. If I'm understanding the question correctly, he's asking what is the site of action inside the mitochondria for the mitochondrial AKT. That's a really good and important question that we are trying now to find out. We reopened recently, so we're getting into that, and I would be happy to share that very, very soon. Yeah.
Thank you, Qutuba. The next question is from Viswani, and the question is whether you have noticed any kind of insulin receptor changes in your stress hearts.
Yeah, another good question. I'm really sorry, guys. We just finished this first series of our heart failure experiment just before the lockdown. So this is all in the process of the analysis. So hopefully we'll be able to tell very, very soon. But we're looking into that, absolutely.
Great. I think some of the questions I might see might be a little bit different. I'm trying to make sure that we're on the same page. The next question I see asks, sorry, is aimed at Qutuba as well. It says, do you analyze for effects of the inhibitors on other classic insulin response pathways, including insulin-dependent glucose transporter GLUT4 in different studies?
Yeah, well, for the series of experiments that we did with the inhibitors, it was acute exposure, and we looked at the expression of GLUT4 and GLUT1 as well, and we didn't see much of a difference with any of the inhibitors. So yeah, we looked into that. We didn't see much of a difference.
I think the questions are a little bit jumbled. That's why it's not in order. I'm sorry. But this question is for Qutuba again, and it's from Eric Weatherford. And he's wondering whether you use separate antibodies for AKT1, 2, or you use the general AKT antibody. Yeah,
no, absolutely. Important question. So for the data that I showed, this is for AKT1 and AKT2. It wasn't specific, but what we're doing now is to go and see which AKT, whether it's AKT1 or AKT2, is actually more important in this, or maybe they are both important. So this is what we're doing right now. No, it's an important question.
Great. The next question that I'm seeing is for you, Qutuba, as well, from Gary Baxter, that asks, the attack model is a pressure overload model. Do you know if the role of AKT in stimulating glucose oxidation might be different in ischemic cardiomyopathy?
Yeah, so hi Gary. So Gary Baxter is my PhD mentor, and it's lovely to see him watching today. So yeah, so we haven't done any work with ischemic cardiomyopathy model, but what we know from experiment is that what we see, what this uncoupling between glycolysis and glucose oxidation exists in, for example, models of ischemia reperfusion during ischemia and during reperfusion, as well as we see as well in different models of diabetic cardiomyopathy. So I would speculate that maybe there is a good potential for this to happen in the ischemic cardiomyopathy as well. But good question.
Sorry, Qutuba, to give you shock. We did not plan this to give you no time to prepare your question, but it just happened. Anyway, the question I see the next is from Konstantinos, and again, it's for you. He's wondering whether the balance between phospho-AKT mediated effect of insulin versus release of fatty acids from the intracellular lipid stores.
Yeah, no. Hi, Constantino. How's it going? So, really good question. So, what I can comment on is the effect of phospho-AKT and what it does to the glucose oxidation. I honestly not aware of the importance of this mechanism in deploying intracellular lipid store, or how it will act, or its role in deploying the lipid store in the heart. But I'm sure you know. So you probably can explain it a little bit, but I'm sorry, I don't know about the fact, but I can speak to the role of AKT in mediating the dark insulin stimulation or glucose oxidation.
Thank you. All right, the next question I'm seeing is for Dr. Wang. It says, as you mentioned, several groups, including our group, demonstrated the importance of O-GlutNAc in hypertrophy development under TAC or ANG2 treatment, inhibiting O-GlutNAc being sufficient to block hypertrophy development. However, we also showed that treatment with OGA inhibitors are excellent O-GlutNAc inducers, but are not sufficient alone to promote cardiac hypertrophy. What is your clue to reconcile these data in your nice paper?
First of all, I want to thank this question, and I'm very familiar with this paper, and we actually talk about in our lab meeting groups. Yes, indeed, but it from our understanding is it's indeed a certain treatment that does not directly lead into hypertrophic growth. We also try to work on why. I think one of the explanation we have is the downstream events is not a general increase of gluconetic modification. It's rather certain players or key players being modified. So if you only look at the increase of gluconetic modification in all cardiac proteins, it may just shadow specific changes. I know it sounds like impossible to understand, but that's what we try to figure out, what particular protein be modified is not, and it's not just the general total increase of gluconetic modification. And I also want to point out is when we treat the cardiomyocyte with gluconetic, we find different concentrations give different effects on cardiac hypertrophy growth. For example, you have a certain concentration, but two concentrations, they all increase gluconetic modification, was only one of those leading to hypertrophy growth. So I think, as I say, possibly due to selective change in specific proteins.
Okay, thank you. The next question is for Qutuba again, and this attendee is wondering whether you notice any kind of GLUT4 changes to your altered AKT signaling.
Yeah, good question. I think I already answered that question. We looked at the translocation of GLUT4 and the expression of it in the normal heart, and we didn't see much of a difference. In terms of heart failure, we haven't done that work yet, but it's a work in progress. So hopefully soon we will be able to tell. Yeah.
Thank you. I'm sorry, I think I dropped off for a minute on the Zoom call and it erased some of the questions on my end, but the next one I'm seeing is for Jiao Wang. that says, congratulations on your work. How about inflammatory status in GFAT1 mice?
We did not measure the inflammatory markers in circulation, but we did was in the transgenic mice, and in knockout mice, we did look at in the heart at protein at the mRNA levels of inflammatory markers. In the transgenic mice, we see the increase of certain biomarkers, and then in the knockout mice, there's a decrease. We didn't put it in the paper, but that's the findings we had. Thanks.
So the next comment is from Thomas, and then it's for Qutuba. So Thomas is wondering, how can you explain your recent data which suggest AKT enhancement is protective in TAC? However, there are a couple of publications from Zumo's lab, as well as Ken Walsh's lab, where they kind of notice the opposite to show physiological hypertrophy and cardiac dysfunction with AKT. How can you explain your present data?
Yeah, no, absolutely. Hi, Thomas. So good question. I think I'm aware of these studies, and they're really, really high quality studies. However, I don't think there is a general consensus about the role or the crosstalk between AKT and mTOR so far, and the literature is just supporting both sides of the arguments, whether AKT activity can trigger or can stimulate mTOR and vice versa. But what I can tell you is that in different models of heart failure that we looked at and we investigated, we often see the improvement in insulin sensitivity, which is often accompanied with an increase in the activity of AKT, is also associated with decrease in mTOR activity. So the question becomes, which isoform of AKT is important to trigger mTOR? And the other important question is, what pocket and what residue of mTOR that can be more important than the other pockets and residues? I think this is a really interesting scope for further investigation. I'm just hoping I didn't get this question for your BCAA paper. It's going to be a difficult one to answer.
Qutuba, sorry, do you mind if I just butt in quickly? I mean, has anybody compared the AKT levels in these different experimental conditions? You know, are you inducing different, levels of changes in AKT expression?
Yeah, that's a really good question, Deborah. So in terms of the mitochondrial translocation in this experiment that I showed, when we stimulate these hearts with insulin, we see an accumulation of AKT inside the mitochondria. So the answer is yes, we see an increase in the accumulation of AKT inside the mitochondria. And we see that this accumulation following insulin stimulation is impaired in the failing heart. So that was the main conclusion from the last study that I showed you. We think that this impairment in mitochondrial classification of AKT into the mitochondria is what playing a major role in impaired insulin-stimulated glucose oxidation in the failing heart. So yeah.
Okay. But you have no evidence of kind of differences in expression, really, or you can't speculate on that, really, can you?
For different isoforms of AKT?
Well, I mean, for different, different models that have been used, like overexpressing models.
No, that is other people work. No, I'm not aware that this was investigated.
I'm just thinking in light of contrasting conclusions, whether that's an explanation.
I think that's a really good idea. No, I agree.
Great, thank you. So the next question I'm seeing is for Dr. Zhao Wang. It's from James, who says, very nice work. Have you observed any association between mTOR change by HBP pathway associated and cardiac autophagy?
That's a very important question. we find we did try to address this by looking at LC3, for example, or Western blot. In our hands, we did not see very obvious change, like a very consistent change from experiment to experiment. So we, to be honest, we don't know the answer, but I guess that's very logical to ask whether activate mTOR might suppress autophagy in this process. One possibility would be at different stage of the whole process, the answer might be different. But from short answer is we do not know. We don't have a conclusive answer right now.
This is a follow-up question from Viswani to Qutuba again. I think you answered the GLUT4 part, but do you think GLUT1 somehow get regulated with your inhibitors?
In terms of GLUT1 with the inhibitors, we didn't see much of a difference in the expression of GLUT4 within this short period of time. What we know from looking at glycolysis is that there was no significant difference in glucose uptake. Otherwise, that will drive glycolysis to increase. So I kind of touched on that very briefly. I'm sorry because of the time, but we don't think we had any significant difference in neither glucose uptake or the pyruvate supply to the mitochondria, as evident by no change in glycolysis, which is the main control here.
Thank you. So the next question I'm seeing is for Dr. O'Shea from June Wave, who asks, does oxy-deoxy-myoglobin affect optical mapping since it will change the absorbance of myocardium?
Yeah, so the answer is yes. So traditionally, the most commonly used dyes within the field have been excited around 530, and then your emissions look at 630 onwards. So that's right in the sort of in the optical window where you have high absorbance by biological chromophores, in particular oxy-deoxyhemoglobin. So what we've seen recently, and what's very exciting, is the production of dyes which are moved towards the infrared and the infrared spectrum. And why it's important is that it allows them for the possibility of doing optical mapping studies without using crystalloid solutions like Krebs solution and so on, but in actually blood perfused systems, and there's not a work that. The answer to that question is, with the dyes that are most commonly used, and even with those near infrared dyes, there will be some change. The answer is yes.
Okay, so the next question is from Rushita to Qutuba. She's wondering, are the underlying mechanism for insulin-stimulated mitoglucose oxidation versus enhancement of glucose uptake and glycolysis mutually exclusive? Or is there a crossover between these processes in healthy versus the diseased myocardium?
Hi, Sheta. Good to see you again virtually. Good question as usual. So that's really important to explain. What we think happening is that these two pathways, whether the direct enhancement of glucose uptake and glycolysis, and thereby feeding more pyruvate into the mitochondria, is working in parallel with the direct insulin stimulation of glucose oxidation in the normal heart. What happens in heart failure is, as I explained in the introduction, we have this uncoupling between glycolysis, which goes up in heart failure, while glucose oxidation is down as an overall mitochondrial oxidative metabolism goes down in heart failure. So we think that we're losing the other part of the metabolic effect of insulin on cardiac energy metabolism, which is the direct effect on stimulating glucose oxidation. So to answer your questions, yes, those are two pathways working together in the normal heart, and we're losing the mitochondrial part, or the direct stimulation of mitochondrial glucose oxidation in the failing heart. And that's what we're trying to restore.
Great. The next question that I see is from Oscar Moreno, who's asking Chris, are you using LED or pulse laser for the stimulation of dyes? Also, I wanted to ask have you noticed a high variability in atrial action potential duration, and how did you manage the problem of spatial averaging of the signals?
Yeah, so to answer the first question, we use LEDs in our specific setups, but other groups also use pulse laser technologies. I think those kind of technologies come more in play when you're looking at more, say, dual dye experiments. We need that fine temporal control, or for example, optogenetic experiments, we have that interplay. The kind of experiments that we've done, it doesn't really, LED illumination works fine. In terms of the actual preparations, we do see high variability in signal quality mainly. So I'd say it's probably linked to that, where you might get changes in action potential duration. I don't see a huge difference in action potential durations. The quality of actual preparations in terms of signal you get tends to be less, just for the very simple idea that you have less tissue degradation, less signal quality. You also have sort of thinner tissue and so on. And then, so the third bit about spatial averaging of the signals, again, yes, obviously you have, if you spatial average, you get the blurring of the signals and so on, which obviously isn't advantageous. Sometimes it's necessary just because of poor quality signals. In general, to try and avoid these problems, you either avoid spatial averaging, or what we try to do is more, when we do apply spatial averaging, apply sort of Gaussian filter averaging, so you get that sort of very sort of fine control, hope you get rid of random noise. But yes, that is an issue with optical mapping. Obviously, we try and get these high resolutions. Ideally in a world, we'll never do spatial averaging, but it's again one of these interplays between data quality versus actually then sort of being relevant to the actual signal you get underlying.
Chris, do you mind if I just butt in quickly and just ask a kind of a follow-up question to that? In your opinion, have you ever seen completely different interpretation of the same data set based on, you know, different sort of processing of the data?
Yeah, so obviously, if you get a massive underlying change, it will be, even if you apply Gaussian field, don't apply field, it will still be there. I think the area we've seen it mostly would be when we looked at sort of change through the upstroke. So we did that study with James, where we looked at carbon dioxide and flecainide, which are known to have change. And what we did within that study is actually look at different temporal and spatial filters. And what you can see is then, these are very subtle changes, but when you apply some filtering methods, you can no longer see these changes. So yes, I think you can. You can have effects where particularly temporal filtering, I find, if you apply the wrong temporal filter, you can really sort of mask the actual physiological changes or the underlying physiological baseline conditions.
So considering then you are releasing, and Igor Efimov is releasing, you know, software for analysis, do you think now in the papers we need to specify in our results sections or in our method sections what filtering we've used, not only what software have we used?
Oh yeah, well, I think most people, well, what I see, most people do, and they definitely should. I think, yeah, because even if it's just in the supplement, it's definitely a very important factor. In particular, if it's optical mapping figure, if you get these raw signals, ideally, I'd like to see raw signals all the time as well. But.
Fine. Thank you.
Thank you, Chris. I think now we kind of catch up with Sarah. If I miss your question, I sincerely apologize in between, but the next question is from Kevin to Qutuba. How does the translocation of phospho-AKT occur? In the heart disease, do you have more translocation issue or AKT activity?
Yeah, that's a really good question. I'm not sure if I have the data to support what I'm going to say, but here's what happens. So we see that this translocation of AKT is impaired in the failing heart. I'm guessing you're asking how this translocation actually happens in the normal heart. That's a really interesting topic that we are trying to look at. I have zero data to do any speculation, so I don't know, but it's a fantastic question. Thank you.
Thanks. The next question is from Rio. It's to Qutuba and Zhao. It is they're asking a more general question. How does glucose transport from the circulation to the cardiomyocyte? Is it uptaken first by endothelial cells and then passed to the cardiomyocyte, or does it go directly to the cardiomyocytes?
Do you want to have a go, Xiao?
Well, I actually not familiar with this. I think, from my understanding, it's to go through the lymph system. I could be wrong.
I'm curious to hear both of your answers here. I don't know.
It's an important question.
That's a better than what I was supposed to. Okay, so I can't comment on what happens at the level of the heart, maybe. So we know that insulin can stimulate glucose uptake by stimulating the translocation of GLUT4, and that's why GLUT4 is called the insulin responsive glucose transporter. But the heart can also take up glucose through GLUT1, which is not insulin dependent in an insulin independent manner. So these are the two main glucose transporter into the heart. Don't ask me what regulates GLUT1 because I don't know. That's why I know about the uptake of glucose into the heart. I hope that answers your question.
This is like a PhD exam question.
Yeah.
I like to find out what.
That's a good, but no, absolutely valid question. I like it.
Yeah, it's a good question. I think we have in Purvis lab, in Philip Sears lab, there's a person working on the lymph system. It seems there's a lot unexplored question there, right there. How the nutrients or hormones from the circulation get into the targeting cells. The short answer is I'm not entirely sure about this. I need to work on those.
Okay, so I have a couple of questions for Chris from the same attendee. And so this person is wondering, there are multiple fluorescent dyes which can able to measure metabolic changes. Is it possible to use the combination of voltage or calcium imaging and metabolic imaging in the cardiac tissue, number one. Second question that this person has is, can your software process dual voltage and calcium imaging?
Yeah, so the answer to the first question is yes. So some of the very earliest optical mass experiments actually employed this exact sort of methodology using metabolic dyes changes with combining them with voltage engines and so on. I have to say I'm not up to speed with the sort of properties of metabolic dyes, but again, the main problem will be, as with when you try to combine optical mapping with object-genetic techniques, with a spectral overlap problem, the idea that can we effectively separate those two signals? But the answer in theory is definitely yes, and it has been done. The second question, we're working on it. It's getting there. It's kind of thing that's been very stop-start for quite a while now. So we have sort of versions that work with, and in principle, everything's there. So you can do it manually within our software at the moment. So basically, analyze a voltage signal independently, measure, reduce the calcium maps as well, and then sort of interplay them with the raw data. But obviously, it's a very elegant solution, and we are working on making it so there will be a specific module which takes those two channels and combines the data. And then also, hopefully, then, if we also do for other dyes, for example, metabolic change and so on. So it's definitely getting there, work in progress. But the answer is kind of at the moment, which is the best answer.
But Chris, this is a really excellent question, actually. And I think we should try this sometimes when we get a chance.
Yeah,
I agree. Induce ischemia locally, see what happens with calcium, as well as, I don't know, like maybe some sort of mitochondrial, fluorescent dye, which will report on mitochondrial activity or something like that. I think.
Yeah, really not to do. Yeah, I think it'd be a great idea. I think in theory it's definitely possible, and I've seen it done before. But you have to read through and go. Obviously, these things often are quite hard to read on yourselves. So we'll work out. But yeah.
Great. The next question is also for you, Dr. O'Shea, from She. They ask, what do you think is the technique limitation for achieving perfect detection of both calcium and APD signal in mouse heart?
Yeah, so there's a few different things going on here. So firstly is just how the dyes interact with each other would be a simple thing. So one of the things that we found is that in preparations, you get really nice loading of action potential dyes individually, really nice calcium dyes individually. Occasionally, when you try to combine them both together, we lose one of the signals for various reasons. We think maybe the saturation with the calcium dye to get the signal, or these sort of interplay. The other things which come from a more technical side are, on the software side, it's not too big an issue. As long as you can effectively separate the two systems, either by the wavelength or another method, for example, dual excitation, then it's pretty simple. Just take the singles, treat them independently, and put them together at the end. What's more of an issue, and again, we ourselves haven't done many experiments, but from speaking to other people who have, seems to be the hardware problem, getting that perfect alignment between the tissue. So you know that one pixel in your voltage image is exactly the same as your pixel in your calcium image. And that seems to be an issue for quite a few people. So that's one of the other sort of technical limitation. But again, it's routinely possible, and there's many groups that do it. So it's definitely possible.
Okay, so the next question, based on the question, I think it is for you, Chris. I'm going to tag the follow-up question to the next question together. So if you can answer both at the same time, that would be great. So can you use your technique, optical mapping, in vivo? That's the first question. I think the same attendee is wondering, do you think more than 2 cameras will reduce the distortion of imaging?
Yeah. So the first question, it's getting there. So originally it was, quite a few of the different things I talked about, for example, need for mechanical uncoupling, the problem with toxic dyes and so on, has traditionally limited to be an ex vivo technique, but there's been some really nice work, well, actually quite a while ago, sort of using strategies such as pulmonary bypass to achieve this sort of more in situ optical mapping. There's been recent work that's combined some of the motion artifact correction techniques with some of the sort of those redshifted dyes that I mentioned, which have achieved these almost, you could say, in situ, well, in vivo optical mapping using fiber array detectors. Particularly Peter Lee has produced quite a lot of really nice work recently on this subject. So it's getting there. It's not sort of as high resolution, for example, an electrical amount art, but it is getting there. Will we ever get to the point where it can be clinically relevant? There is sort of 1 or 2 FDA approved optical voltage dyes and so on, but that's a long, long way still away from actually becoming possible. For the second question, so it depends what sort of distortion the attendee is actually thinking about. In terms of mechanical uncoupling, so if you have a heart that's still beating, it could help slightly because you get those two camera views, so you can see which way the heart's moving. Then you can apply these motion, these computer vision techniques. If you talk about distortion just in terms of other artifacts, for example, things like optical blurring and so on, what you need to do really in terms of sort of the 3D architecture is combine sort of surface capture software as well. Our algorithms actually very detailed to capture software. So having more cameras does help that and make that easier, but it's still not a trivial problem. And in particular, Eagle FMOS group has, about a couple of years ago now, released sort of suite of open source software platforms for this exact reason, because it's really hard computational problem, even with 4 cameras. So I look at each view. To combine them all together and get true 3D optical mapping, it's still pretty difficult.
Thank you. The next question is for Dr. Wang from ASMA. They ask, did you check some common readouts of autophagy like LC3 and Becklin-1?
Oh, yeah, I mean, he also followed up with that. We did, like the question I addressed previously, we look at LC3. We didn't look at a Becklin-1, but LC3, as I said, we don't have a very consistent and convincing change across different experiments. So, we think a short answer is we don't think it's strongly changed it.
Okay, in my screen, I don't see any more questions. So let's, Sarah and I,
sorry, there's one more question in the chat from what I can see.
Oh.
Can you see it from Wahiba Dari?
Chat. Can you please ask? Because I was chatting too much.
I do see it. So it sounds like it's for Dr. O'Shea. They say, I'm using optical mapping to image synchronously engrafted cardiomyocytes expressing autonomous voltage sensors with the guinea pig host myocardium dilated. If activation map analysis is done separately for each channel, would it be possible to combine data from both to see if the graft is coupled or not?
Yeah, so I believe so, yes. You'd have to, the problem with combining different channels like that is if you have an effective reference then to basically to stitch those signals together. I believe, yes, without sort of seeing the actual intricacies of this experiment, it sounds like a very complicated, so I'd be not going there, but I don't see why that shouldn't be possible.
But that should be probably the same issue like the voltage calcium,
yeah,
right?
Exactly. As long as you have an appropriate reference, for example, I don't know how you, for example, you're stimulating these preparations, but if you have something to reference, so you can make sure. One of the problems if you analyze two things separately, and for example, our software, which is you're basically looking at the actual intrinsic signal within, for example, the optical voltage signal as a time reference. But so then we're doing that separately with two different channels. You need to make sure there's a common channel or common stimulus in both channels where you can effectively line things. So that would be the only sort of limiting factor in doing that. But that should be routinely possible from what you say.
But Wahiba, I mean, I would just say that you may want to just contact Chris. I don't know if Chris, you have time, but you know, he may be able to help because I know that Chris, you've dealt with a few issues like that so far, but similar.
We've always found a way around it. So.
Okay, same goes to every other questions. If you want to email Qutuba or Zhao directly, please ask your question. I don't think any more question at this time. So Sarah may conclude the session.
Thank you again for everyone that spoke today and for those that attended and asked questions. We're grateful for everyone's contribution today. And just wanted to again thank everyone and remind you these are a continuing series to join in for another time. Thank you.
Thank you very much for taking care of us. And
Thank you.
For everything. Thanks
You
a
guys,
lot.
well done. Great talk.
Good to see you guys.
Thank you.
Bye-bye.
Bye. Bye, everyone.
Bye-bye. Thank you.
Bye-bye.